I chose to look at this paper by Kant and Miner, which looked at the courtship and mating behaviors of the cabbage aphid parasitoid D. rapi. D. rapi is a species of parasitoid wasp that attacks several species of aphid, including the agricultural pest B. brassicae. This makes it a valuable species for biological control in agriculture. All hymenopterans, including D. rapi, are haplodiploid. This means that males are haploid and hatch from unfertilized eggs, whereas the females are diploid and hatch from fertilized eggs. This is important because only females can be used in pest control, as only the females oviposit into the aphids. The authors predicted that male parasitoids will display courtship behaviors, and that these courtship behaviors will induce receptivity in the females. They also predicted that females that mate with already mated or sperm depleted males will be more likely to remate. The authors collected D. rapi and B. brassicae from a cauliflower field in New Zealand. The insects were then maintained at about 20 degrees Celsius, 60 to 70% relative humidity, and a 16-hour photophase. The authors only used D. rapi that emerged from 6 to 7-day-old aphids in order to keep the wasps at a consistent size. Each wasp was also given a 10% honey solution before the mating experiments. In their first experiment, the authors wanted to investigate at what age D. rapi becomes sexually receptive. To do so, they grouped the wasps by age into 1, 3, 6, or 24 hours old. Each individual was then paired with a 24-hour-old individual of the other sex. Each individual was only used in one receptivity experiment, so all individuals used were unmated. 30 to 37 individuals were used per age group. The authors recorded the behavior of the individuals until the pair mated. If the pair did not mate, they were considered to be unreceptive and were not used in the statistical analysis. The authors also wanted to look at the courtship and mating behaviors of D. rapi. To do this, they fitted a microscope with a video camera to record behavior. 24-hour-old males were first observed for 60 minutes on their own for a baseline idea of behaviors, and then for 60 minutes after pairing with a female. If the pair did not mate, they were considered unreceptive and, again, not included in the analysis. The authors also recorded the length of the pre-mating and copulation times. Finally, the authors wanted to see whether or not D. rapi would mate multiple times. To do this, they selected some pairs that had successfully mated in a previous experiment. These wasps were rested for 60 minutes and fed a 10% honey solution. Each individual was then paired again with a 24-hour-old unmated individual of the opposite sex and was again observed for 60 minutes. This procedure was repeated 8 times for each original male and female. In their sexual receptivity experiment, the authors found that mating attempts were made sooner by male than female parasitoids. They also found that mating success was significantly lower in younger females than in older females, but this pattern was not true in males. As you can see in the graph, mating success rose with female age, but stayed relatively consistent between the male age groups. Before I get into the mating behavior results, I wanted to quickly show you how the authors defined the different behaviors they observed. Grooming was when the wasp would rub their legs or antennae together while standing. Approaching was when one wasp would move closer to the other, and encountering was when the male and female wasps actually made physical contact. They defined male courtship behavior as when the male would briefly move his wings up and down, frequently while approaching the female. They defined mounting as when more than half of the male's body is on the female, and copulation as when the male inserts the adagus into the female, the adagus being the reproductive instrument that male insects have. They also observed antennation, where the male would move his antenna vertically during copulation. In their courtship and mating experiment, they found that when placed together, males and females would pretty immediately begin waving their antenna. This may be to pick up on pheromone signals put out by the other individual. The first encounter would occur about 5 minutes after pairing. 91% of males displayed the courtship behavior I just defined on or just before the encounter, and males also approached females more frequently than females approached males. Some females would initially avoid mating by leaving the encounter site, but then 13% of these females would later mate with the same male they had initially rejected. The males were also persistent, displaying courtship behaviors until mating occurred. And in 75% of cases, these courtship behaviors did lead to mating. This is an ethogram the authors made to show the order of behaviors they observed. As you can see, it also shows how many males made it to each stage. So they started with 37 pairs, and in the end, 26 of the males successfully mated. You might see where the female can walk away in order to prevent mating. The authors also saw, as I mentioned before, the males would continue courtship until mating occurred, so longer mating times were associated with higher numbers of courtship displays. However, the frequency of male courtship displays would decrease after female rejection. Um, so the total number of courtship displays would level out after, as you can see here on this graph. In their multiple mating experiment, the authors found that female D. repi would become unreceptive after copulation and would not mate for a second time, regardless of the mating status of the male they initially mated with. 56% of males, however, displayed courtship to these unreceptive females that had already mated, and 20% attempted to mount these females. Overall, the authors found that male D. repi do display active courtship behaviors as well as repeated mating attempts. Female D. repi mate only once, but are selective about the male they mate with. This is important because, as I mentioned before, D. repi can be very useful in the biocontrol of the cabbage aphid, which is a major agricultural pest. However, as I mentioned before, females must successfully mate in order to produce more females, since daughters are diploid, and only female wasps are able to control cabbage aphid populations, as only female wasps will oviposit into the aphids. Overall, the results of these papers may be very useful in helping production of D. repi for agricultural pest control.